This is Teacher Doctor once again, Teacher Doctor MUGOMBA Ronald, and I'm back again, and today we are going to go ahead and look at movement in and out of the cell. We are going to look at the different processes which are involved when we are moving materials in and out of the cell. So, substances usually are transported for, we ask ourselves such a question. In this video, we are going to look at the different processes that are involved during movement of materials in and out of the cell. That is to say, we shall mention them. We shall look at why we need to move materials in and out of the cell. We shall look at the different groupings of the different processes involved in movement in and out of the cell. So, the first question that we shall start with is, why do we move materials in and out of the cell? That is the question that we'll start with. We need to move materials in and out of the cell to facilitate chemical reactions. We need to move materials in and out of the cell to maintain the electrical potentials at the cell membrane, which are very important when it comes to transmission of nervous impulses. Chemical reactions like photosynthesis, chemical reactions like respiration have to take place because we have moved oxygen in and taken out carbon dioxide. We need to move materials so that we can eliminate wastes from the cells, because there are a number of wastes that are produced from the cells that we need to eliminate. So you need to move materials in and out of the cell so that you can go ahead and eliminate those wastes. So when you look at the whole idea, we begin with the cell membrane. So the cell membrane is usually surrounding the entire cell. So it's like the barrier across which materials will have to be moving in and out of the cell. It also covers the different cell organelles. It is usually fluid in nature. And what we called fluid in nature, we say it is fluid simply because it is in constant motion, or it has molecules that are always moving. It is made up of predominantly of a phospholipid bilayer, with the head being polar or hydrophilic and the tail being nonpolar, that is, say, hydrophobic. So the tails usually project inwards towards each other, and they create what we usually call the hydrophobic region. And the heads usually project outwards and inwards to the cytoplasm because they are hydrophilic, they are water-loving. It also contains some proteins within it, and these proteins are in categories. We have those ones which are intrinsic or integral proteins, which act as carriers and channel proteins. We have those ones which are known as peripheral or receptor proteins, which can act as antigens, which can act as receptors, which can act as carriers. So you can see the peripheral proteins, like the acyarins. You can see them there. We have the integral proteins. For them, they span through the cell membrane. And we have cytoskeleton, like the spectrins. The cytoskeleton are very important when it comes to maintaining the shape of the cell, ensuring that the cell organelles are anchored very well. So that is the cytoskeleton. So you can see that is the structure of the cell membrane according to what they usually call the fluid mosaic model. You can see the extrinsic proteins, you can see the glycoproteins, you can see the glycolipids. So this is the extracellular fluid. Intracellular fluid is the fluid that is found within the cell, that is the cytoplasm. You can see that the membrane is made up of two layers, the bilayer. You can see the hydrophobic tails, they are here projecting towards each other. And you can see clearly that the hydrophilic heads are projecting out inwards into the ECF, inwards into the ICF, and outwards into the ECF. You can see that we have molecules known as the cholesterol that are in between the phospholipids. You can see, so that is basically the what they call the fluid mosaic model of the cell membrane. So the cell membrane performs a number of functions, as we are going to outline them. So it can act as a semi-permeable membrane or barrier. It maintains differences. When you look at it, it is very important in maintaining difference in composition between the intracellular fluid, that is, say, the cytoplasm, and the extracellular fluid, that is, say, the surrounding cell of the cells, surrounding solution of the cells. It protects cells from toxic substances. It is very important when it comes to getting rid of waste materials. It transports nutrients to the outside of the cells. As we saw previously, we saw that the cell membrane has receptors which are very important for receiving chemical signals, and some of which are very important for receiving electrical signals, so that we can see communication among cells. So we can go ahead and look at transport across the cell membrane. When you look at transport across the cell membrane, it is broadly divided into two categories. We have what they usually call passive transport and what we usually call active transport. As we are going to see, we have what in passive transport, this is the type of transport which moves materials along their concentration gradient, like simple diffusion and facilitated diffusion, and sometimes filtration and osmosis. When you look at active transport, we have two categories. We have primary active transport and secondary active transport. We have endocytosis and exocytosis, what they commonly call bulk transport. Now let us go ahead and look at the different methods of transport in details. When you look at, we have passive and active. We have seen the different categorizations, that in passive we have diffusion, and diffusion can either be simple or facilitated. In diffusion, we usually see the lipid bilayer playing the most important role, the simple diffusion, and the protein channels. So we have what we usually call the leaky calcium channels, the leaky sodium channels, the gated sodium gated channels. They are very important in allowing materials to cross the membrane with ease. Qualities of those materials that cross the membrane with ease: they're usually very small in size, and they're usually non-polar, because the cell membrane is non-polar as well. And then we have facilitated diffusion, we have osmosis, and we have filtration. Now, in simple diffusion, molecules move from a region of high concentration to a region of low concentration until equilibrium is reached. Equilibrium point is reached when the concentration of the molecule that is moving on either sides of the membrane is the same. Like diffusion of oxygen molecules, so you can see these atoms, their molecules are moving until on either sides of the membrane they are the same. You can clearly see that. You can see them moving. They move from where they are more concentrated to where they are less concentrated until equilibrium is reached. Okay, so you can see clearly they move until equilibrium is reached. Okay, so now let's go ahead. Now when you look at simple diffusion, it takes place through the cell membrane. Through the cell membrane, the lipid soluble substances like oxygen, carbon dioxide, alcohols, and steroids, they can cross with ease. Lipid insoluble molecules like water, like urea, like sugar, they are less permeable, so they cannot cross with ease. They are less permeable. Electrolytes and charged molecules cannot cross. Electrolytes, that is to say sodium ions, chloride ions, they cannot move with ease through simple diffusion. Any mutation that can take place on any of those carrier proteins. So you can look at the carrier proteins. The carrier proteins, for them they come in to play a role when it comes to facilitated diffusion. So the electrolytes which cannot cross with ease across the cell membrane, they will have to cross the cell membrane through special channels known as carrier proteins. So these special channels are going to carry the molecules, the electrolytes, across the cell membrane, like the leaky sodium channels, like the leaky potassium channels. They are very important. We also have the gated channels. They open in specific conditions. When you have a lot of sodium ions accumulating, then we see opening, then that one is called sodium gated channel. We have very many of them, so they open in response to certain ions. When they are present, they open and shunt them across. So in facilitated diffusion, we usually see molecules will cross the cell membrane. They usually cross the cell membrane because these channels usually provide charged areas across which they can cross. These materials can cross which are charged because the cell membrane is predominantly non-polar. So we have a number of factors that affect the rate of diffusion, like the solubility and lipids. Lipid soluble molecules will usually cross the cell membrane with ease, unlike the lipid insoluble molecules. Molecules which are relatively small in size will cross the cell membrane with ease compared to molecules which are large in size. At a higher temperature, molecules will possess a higher kinetic energy, so they are most likely to cross the cell membrane with ease as compared to molecules when it is at low temperatures where they have low kinetic energy. The thickness of the membrane, when the membrane is relatively thick, the rate of diffusion will be lower. The surface area, you can see that, for instance, the alveoli have a very large surface area. We shall have a faster rate of diffusion in an area with a higher surface area than in an area with a low surface area. So when you look at other factors like the concentration difference, the concentration gradient or the slope, how different are the concentrations on either sides of the membrane? If the concentration difference is very high, then we shall have a very fast rate of diffusion. The pressure gradient and the electrical gradient, all those are factors that affect the rate of diffusion. It is important for us to note and look at this principle known as Ficky's law. Ficky's law tries to look at the rate of diffusion and put up all these other parameters which affect the rate of diffusion. So the net rate of diffusion can be given from, it is usually proportional to the concentration gradient of the substance. It is usually proportional to the product of the concentration difference of the molecules, the permeability of the membrane, and the surface area of the membrane. So it is rate of diffusion is a product of concentration difference, permeability of the membrane, and surface area of the membrane. So that is Ficky's law. It is very important when we are looking at the process of diffusion. Facilitated diffusion involves movement of materials which are larger in size, as I've told you about them. They are water soluble. It is also passive because it moves materials along their concentration gradient, but it uses carriers to mediate its process, the movement of materials. The materials come and bind on a carrier, and the carrier then takes up the materials. You can see from here, this molecule has bound on the carrier, and now the carrier undergoes a conformational change to shunt the molecule across the cell membrane. That is facilitated diffusion. But it should be noted with maximum emphasis that it is passive and it does not use any form of energy, despite the fact that we are seeing conformational change happening in the carrier. So you can see what is happening. So there are a number of things that we need to note when we are looking at carrier-mediated transport or facilitated diffusion. In simple diffusion, molecules were moving with less specificity, but in carrier-mediated transport, materials usually move with some kind of specificity. So these carrier proteins usually, they are specific in what they carry across the cell membrane. Now, there is also some kind of competitive inhibition. Certain times they can compete. Some kind of competitive and non-competitive inhibition, and then there is what we call saturation. These carrier proteins can sometimes become fully saturated with a certain molecule, and they are unable to carry molecules across the cell membrane, so they reach equilibrium point. So sometimes we can block the receptors, like it is used in pharmacology when we are looking at drugs. So we can use certain receptors to block certain materials from crossing the cell membrane. Examples of those carriers include the glucose channels, amino acid channels, galactose channels. They usually in peripheral cells we have the counter transporters of chloride and bicarbonate ions in the renal tubules, etc. Now we can go ahead and look at osmosis. Having looked at diffusion, osmosis is another method of passive transport. In osmosis, when two solutions of different concentrations are separated by a semi-permeable membrane, then we shall see a semi-permeable membrane is a membrane that is impermeable to salts or solutes but permeable to water. Water molecules will freely move from the solution which is more dilute to a solution which is concentrated. So they will move from a less concentrated solution to a more concentrated solution, as you can see from here. So you can see that these molecules, water molecules, will move from the area where we are having a higher concentration of solids, and that explains why you can see that now this volume of the water this side, which had more solids, has increased. Why? Because these solids tend to drag water towards where they are. So you can see that clearly. So osmosis involves movement of water molecules along a water potential gradient from where the solution is dilute to where the solution is more concentrated. So osmotic pressure is like the minimum pressure that you must apply so that you can that must be applied on the solution with a high concentration to prevent osmosis from taking place. And usually it depends on the concentration of dissolved salts rather than the type of the particles, and it is usually expressed in milliosmoles. So the osmotic pressure is that pressure that you must exert on the side where we are having a very high concentration to go against osmotic influx of water into that solution. That is osmotic pressure. So when you look at the types of solutions, we usually divide solutions into three different types depending on their properties in terms of osmolarity. Now, we have solutions known as isotonic solutions, we have hypertonic solutions, and we have hypotonic solutions. Now, if we are looking at isotonic solution, an isotonic solution is a fluid having osmolarity which is the same as blood plasma. That's when we shall say it is isotonic, because iso means the same. So a solution that has the same concentration as blood plasma, we call that isotonic. Or that is having a same concentration as red blood cells, that is isotonic. And it is usually about 0.9 sodium chloride and 5% glucose. Hypotonic solutions will cause red blood cells to swell and hemolyze or burst. Why? Because they have a lower concentration of solids like distilled water. So water will move from them into the cells and causes those cells to burst. Hypertonic solution will cause the red blood cells to shrink. That is to say, to reduce in size. Why? Because the solution has a very high concentration of salts compared to water, compared to the red blood cells. So the cells will lose water by osmosis. Now, so having looked at the different forms of passive transport, let's go ahead and look at the different forms of active transport as well. In active transport, we usually see movement of materials against their concentration gradient. So we have primary active transport and secondary active transport. We also have endocytosis that can include pinocytosis and phagocytosis. We have exocytosis as well. Some things to note about active transport. One, it is carrier-mediated transport, similar to facilitated diffusion. However, the rate of it has rapid transport of materials, and it moves materials against their concentration gradient, that is to say up the hill, and it involves expenditure of energy. This energy is obtained from the breakdown of ATP in the presence of ATPase enzyme. So these carrier proteins are usually specific and they undergo saturation. This can also be affected by competitive inhibition or blocking. Substances which are usually transported in these active transport mechanisms, there are very many, including sodium, chloride, hydrogen ions, chloride ions, iodide ions, sometimes glucose, sometimes amino acids, all those molecules. So when you look at primary active transport, in primary active transport, what we usually talk about is the fact that we have examples like the sodium-potassium pump, we have the calcium pump. The sodium-potassium pump is found almost in all cells. Calcium pump, the one that is found in most of the cells, but so much concentrated in the muscle cells. The proton potassium pump, that is also found there. So usually these pumps are usually driven by energy, and this energy is obtained from the hydrolysis of ATP molecules in the influence of an enzyme known as ATPase. We can say ATP synthase because it is splitting them. This ATP is generated in the mitochondrial membrane, inner membrane, under the influence of an enzyme known as ATPase. So after this ATP has been hydrolyzed, it releases energy, which is going to be used to transport these molecules across the cell membrane. So you can clearly see that in the sodium-potassium pump, we usually enjoy energy. We use energy. So usually 2 potassium ions attached to the outermost part of the membrane, and the innermost part of the membrane 3 sodium attached. So in that way, the membrane ATPase, the carrier then is involved in hydrolysis of ATP, which produces energy, and that energy is important for this pump to undergo a conformational change that is going to cause us to pump 2 potassium ions into the cell and pump out 3 sodium ions out of the cell. So we shall have efflux of 3 sodium and influx of 2 potassium ions into the cell. In so doing, we shall create a very high concentration of potassium ions inside the cell and the negativity of the inside of the cell. And then we shall also create a very high concentration of sodium ions outside. A lot of potassium ions inside, more sodium ions outside. That is very important in ensuring that the cell volume is maintained. So you can see that the pump undergoes a conformational change to shunt 3 sodium inside and shunt 2 potassium outside. Sorry, to shunt 3 sodium outside and 2 potassium inside. We have a number of regulators of the sodium-potassium pump. Like in case you have increased amounts of glucose in your blood, the sodium-potassium pump is switched on so that you can take in more glucose to produce more energy. So that this sodium-potassium pump uses most of the energy that you use in your that is stored in your food. Most of the energy is used for pumping of these ions because they are very important. We need to establish a concentration gradient upon which we are going to move other molecules across the cell membrane. Now, having looked at primary active transport, let us go ahead and look at secondary active transport. Now, in secondary active transport depends upon concentration gradient of the sodium molecules. So, usually, in the primary active transport, we are establishing a concentration gradient. So after the concentration gradient has been established, this concentration gradient is going to be used to drive secondary active transport. It indirectly uses ATP. That is why they call it secondary active transport. Take an example. Because now we are having a very high concentration of sodium ions outside the cell because of the action of the sodium-potassium pump. So now, in case we want to move glucose into the cell, we can couple it to sodium ions. As sodium ions now move by diffusion, simple diffusion from the outside of the cell to the inside of the cell. When they are moving by simple diffusion through special channels, then that same channel can also be a channel that will allow also movement of glucose molecules. So it allows, so sodium is moving along its concentration gradient, but glucose is moving against its concentration gradient. Why? Because the cell usually has a higher concentration of glucose compared to the outside. So as the sodium is moving along its concentration gradient from the outside of the cell to the inside of the cell, glucose also follows against its concentration gradient, where it is more concentrated in the cell, yet it is moving from where it is less concentrated outside the cell. That is what we call secondary active transport. The carriers do not use ATP directly. Yes, they use ATP, but not directly. It is not correct for you to say that they do not use ATP. They use ATP, but indirectly. As I've told you, that they are driven by the concentration differences which were generated by the primary active transport. I've given you an example. Now there are other examples like the reabsorption of glucose and amino acid in the proximal convoluted tubule cells. There, as we secrete hydrogens into the tubular epithelium, we take in sodium, we absorb sodium and glucose at the same time. So such kind is known as, so you can clearly see it here. So you can see here that as we are taking in sodium, we couple the taking in of sodium as sodium is entering. At the same time, along its concentration difference, glucose is also entering. You can see that. So glucose it depends on the concentration difference of sodium. So as you take in sodium by simple diffusion, then glucose also follows. That is secondary active transport, like the carrier-mediated transporter known as the sodium glucose transporter. That's an example. So when you look at the different transporters that we have in our body cells, there are very many different categories. We have the uniport transporters, we have the symporters, we have the antiporters. Now, when you look at the symporters, the uniporters, for them, they transport only one molecule across the cell membrane. That is to say, they can be transporting glucose across the cell membrane into the cell. That is symporter. That is uniporter. Then symporter, take an example of the previous example here. The sodium glucose co-transporter. Now, that one is a symporter. Why? Because it is transporting sodium into the cell and at the same time glucose into the cell. So since it is transporting glucose into the cell and sodium into the cell, then it becomes a symporter. Now, when you look at the other one that we looked at, another transporter in secondary active transport, here in primary active transport, where we are seeing 3 sodium inside out of the cell and 2 potassium inside the cell. That one is transporting sodium and potassium in different directions. This one here qualifies to be called an antiporter. So antiporter, that one in opposite direction, 2 different molecules in opposite direction. Symporter, 2 different molecules but in the same direction. Uniporter, 1 molecule in one direction. So we are going to go ahead and conclude our discussion by looking at endocytosis. So endocytosis has very many different categories. We have pinocytosis, and this one they usually commonly called. So when the cell is taking in materials by formation of an invagination on the cell membrane, so that it can take in materials, bulk transport of materials across the cell membrane. It can either be pinocytosis that exists among all cells. In pinocytosis, we usually form small vesicles to take up liquid molecules into the cell. An invagination is formed on the cell membrane, and then it engulfs small liquids, droplets into the cell. Then we have what we call receptor-mediated endocytosis, like the one that is found in the skeletal muscle fibers. Then we have phagocytosis, that is characteristic of the white blood cells, which usually take up microorganisms like bacteria. When they are eating up the bacteria, they do what we call phagocytosis, solid feeding. The pinocytosis is like drinking, then phagocytosis is like eating. So in pinocytosis, I've told you, we see the membrane deforming, coat protein. You can see small molecules, and invagination is formed, and the small molecules then taken up. That is pinocytosis. An endocytic visco is formed, and so on and so forth. In receptor-mediated endocytosis, it is a selective process which imports important specific large molecules, and it requires energy and the calcium. So calcium acts as a bridge between, it acts as a bridge to bridge the endocytotic viscose and the cell membrane. So when a molecule binds to the receptor, the calcium enables the formation of an invagination that is going to go ahead. Yeah, you can see it there. You can see an invagination has been formed, and then an endosome has been formed, which has the virus, like for the virus hepatitis B, the AIDS virus, all of them are eaten like that, and even excess iron. So in phagocytosis, it is relatively internalizing large multimolecular particles like the bacteria. Certain types of white blood cells are professional phagocytes. I've talked about that earlier. The cell membrane makes contact with this, the material, and then it forms an invagination, and then it's like the bacterium that you are seeing in this picture here that comes, and then an invagination is formed and it is eaten. A pseudopod is formed, it is eaten. We form an endosome, where the endosome is now going to be worked on. The lysosomes will come and bind to the endosomes, and then they start breaking it down. Then finally, exocytosis follows after digestion. Yeah, so something like that. So that is basically movement in and out of the cell. I remain teacher Dr. MUGOMBA Ronald. Consider to subscribe, like, and share. That is the only way you can support, so that we can continue making for you videos to simplify your life. Till next time, have a good day.